Good morning, everyone. I am Valentina Citi from the Department of Pharmacy of the University of Pisa, and I'm going to present some results about the cardiovascular effects of some isothiocyanate and hydrogen sulfide releasing agents, in particular showing their cardioprotective effect. The ischemia reperfusion injury is due mainly to coronary occlusion, so the underlying tissues can't receive the oxygens and nutrients from the blood. During this event, we have the ischemia, the phase of ischemia, in which we don't have oxygen nutrients. After that, we have the reperfusion phase in which the occlusion has been resolved. In this phase, all the nutrients, all the oxygen will reperfuse the tissues, and we have an increase of oxygen that leads to the formation of the reactive oxygen species. Because of that, the damage for the tissue, here we can see the white areas. The death of the tissue is due to also for the necrosis events, so the cells undergo to necrosis mainly during the ischemia phase, and then we have the apoptosis that is mainly due to the reperfusion phase for the oxygen inflate. Hydrogen sulfide has been always considered only a toxic pollutant gas, but in the last few years, many studies demonstrated that hydrogen sulfide is also an endogenous gaseous mediator that is mainly produced due to the enzymatic routes, for example, due to the cystathionine gamma lyase and cystathionine beta synthase, starting from the metabolism of L-cysteine, so an amino acid. So we have a lot of papers that demonstrated that hydrogen sulfide mediates cardioprotective effect due to a plethora of beneficial effects. For example, hydrogen sulfide is a well-known antioxidant molecule. So we can exploit this gaseous molecule to scavenge the reactive oxygen species that characterize the reperfusion phase. We have also an effect on the monocytes, adhesion and infiltration in the endothelium. Indeed, hydrogen sulfide prevents the infiltration of leukocytes. We have many effects on the angiogenesis. Indeed, hydrogen sulfide stimulates the formation of novel vessels. Furthermore, hydrogen sulfide mediates vasodilation. Indeed, it can activate the potassium channels, for example, ATP channels, Kv7 channels expressed in the cell membrane, smooth muscle cell membrane, and in the cardiomyocyte membrane. Activating the potassium channel, we have hyperpolarization of the cell membrane, and we have vasodilation. So, for example, in the case of an occlusion, a coronary occlusion, an early vasodilation can lower the formation of the reactive oxygen species during the reperfusion phase. And furthermore, hydrogen sulfide is also able to activate the metopotassium channels expressed in the mitochondrial membrane. And in this case, we have less apoptosis when these channels are activated if we have hydrogen sulfide. But hydrogen sulfide is a gaseous molecule, so we can't administrate directly hydrogen sulfide because we have risk of overdose. Furthermore, many hydrogen sulfide donors, many salts, have been developed and have been studied, for example, the hydrogen sulfide sodium or calcium, but we have a rapid formation of hydrogen sulfide followed by the rapid elimination rate. So if we think to the clinical practice, we can't administrate this kind of hydrogen sulfide donor. The hydrogen sulfide donors should be low-releasing molecules, so we can have a long-lasting kinetic of the release of hydrogen sulfide, and we can control the dosage of the hydrogen sulfide released into the body. In the last few years, we have demonstrated that the isothiocyanate can release hydrogen sulfide. The isothiocyanate are maybe the most known isothiocyanate are those contained in many edible plants, for example, broccoli, rocket salad, cabbage. So starting from this point, because many studies demonstrated the beneficial effects of isothiocyanate, we wanted to evaluate if these compounds were able to donate hydrogen sulfide. And we have published many papers about that, in which we demonstrated that isothiocyanate, both the synthetic one and both the natural one, can release hydrogen sulfide and mediate a plethora of beneficial effects in the cardiovascular system, for example, antihypertensive effect, anti-tumoral effect, or cardioprotection after the ischemia reperfusion injury. After that, in this work, we wanted to find other molecules to be better isothiocyanate to be administered, and we have started from in silico analysis of a variety aryl isothiocyanate, starting from the H2S releasing rate, solubility, molecular weight, and ADMET profile. So after this in silico analysis, we selected one isothiocyanate. This is a pyridine derivative, as we can see in the picture, and it has good solubility, good hydrogen sulfide release, as we will see after that, and meet all the ADMET rules. Indeed, this isothiocyanate that we called Isothio 25, we saw that it is able to release hydrogen sulfide into the cells. For measuring the hydrogen sulfide release into the cells, we incubated the cardiomyoblast with a dye, that is the WSP1. Here is the structure. And we can see here from this graph that the Isothio 25 is able to release hydrogen sulfide in a concentration-dependent manner. Here we can see also the picture of the fluorescence formed after the hydrogen sulfide release. We selected the diallyl disulfide as a hydrogen sulfide donor well known, derived from the allin sativum. After that, we wanted to study the effect of this compound on the coronary flow in perfused rat hearts, and we can see we administer angiotensin II to induce the coronary spasm that evoke a reduced flow, here about 20% less compared to the basal one. After that, we administered directly to the Langendorf apparatus the Isothio 25, starting from 1 micromolar to 30 micromolar. And we can see that those concentration effect on the cardiovascular parameters, in particular to the cardiovascular flow. Here we wanted to report also the data of another isothiocyanate, the 4-carboxyphenyl isothiocyanate, and we can see that we have comparable results. After that, we wanted also to study the cardioprotection in vitro after the ischemia reperfusion injury. So the rats were treated with isothiocyanate or with a blocker of mitoc ATP, is the 5-hydroxydecanoid acid. After 20 minutes of the blocker, we treated the rats for 2 hours, and then we studied the ischemia reperfusion injury into the apparatus, lung apparatus. And here we studied the area of the ischemia tissue, and we can see from this graph that the administration of increasing dosage of azothiocyanates 25 leads to an improvement of the cardiovascular tissue. So we have a less necrotic area, less white area that is underlined with a TCC dye. And we can see that administering the blocker of vitro-KTP, we have a total abolishment of the cardioprotective effect. This cardioprotective effect was also confirmed by measuring the lactate dehydrogenase, that is the gold marker of the ischemia events, and we have similar results. So we have a total, a total protection of the isothiocyanate at the concentration of 6.7 milligram per kilo, and we have then an abolishment of the lactate dehydrogenase in the presence of the methyl-KTP blocker. Finally, the cardioprotection in vivo ischemia reperfusion injury using the LAD occlusion. So in vivo, we occlude the left anterior coronary, and we can see that the rats treated with the azothiocyanates 25, they exerted a cardioprotection effect of about the 20% when compared to the control one. Thank you for attention, and we can conclude that azothiocyanates can be exploited for the cardiovascular beneficial effect, in particular for the cardioprotection effect. And I wanted to thank you, my professor, Professor Calderone, that always has a good idea about that.